Okay, 13.3, we're going to talk about mutations. So the sequence of DNA bases are like letters of a coded message. So what would happen if a few of those letters changed accidentally, altering the message? What effects would you predict such changes to have on genes and the polypeptides for which they code? So what are mutations? Mutations are heritable, it's a hard one to say, heritable changes in genetic information. So now and then cells make mistakes in copying their own DNA by inserting the wrong base or even skipping a base as a strand is put together. These variations are called mutations, and that's from a Latin word, mutare, which means to change. So mutations are heritable changes in genetic information. All types of mutations fall into two basic categories. Those that produce changes in a single gene are known as gene mutations, and those that produce changes in whole chromosomes are known as chromosomal mutations. So the mutations that involve changes in one or few nucleotides are known as point mutations because they occur at a single point in the DNA sequence. They generally occur during replication. So if a gene in one cell is altered, the alteration can be passed on to every cell that develops from the original one. So point mutations include substitutions, insertions, and deletions. You'll need to look at the pictures in your book. In a substitution, one base is changed to a different base. Substitutions usually affect no more than just one single amino acid, and sometimes they have no effect at all when that happens. In one example, in the base cytosine, if it is replaced by the base thymine, resulting in a change in the messenger RNA codon from arginine to histidine, that's the change that it will make. But a change in the last base of the codon, usually from CGU or CGA, for example, would still specify the amino acidic arginine. Okay, insertions and deletions are point mutations in which one base is inserted or removed from the DNA sequence. So if a nucleotide is added or deleted, the bases are still read in groups of 3. But now those groupings shift in every codon that follows the mutation. So insertions and deletions are also called frame shift because they will shift the reading of the frame of the genetic message. So frame shift mutations can change every amino acid that follows the point of the mutation, and it can alter a protein so much that it's unable to perform its normal functions. All right, now let's look at chromosomal mutations. They involve changes in the number or the structure of chromosomes. So these mutations can change the location of genes or chromosomes and can even change the number of copies of some genes. There are 4 types of chromosomal mutations: deletion, duplication, inversion, and translocation. So deletion involves the loss of all or part of a chromosome. Duplication produces an exact copy, which is what you would think with duplication, of all or part of a chromosome. And then inversion reverses the direction of parts of a chromosome. So if it was BC, it might be inverted to CB instead. So translocation occurs when part of one chromosome breaks off and attaches to another. Okay, just listen to the word translocation. That makes sense that it would break off and attach to another one. So how do mutations affect genes? Well, the effects of mutation on genes vary widely. Some have little or no effect, and some produce beneficial variations. Some negatively disrupt gene function. So mutations often produce proteins with new or altered functions that can be useful to organisms in different or changing environments. So genetic material can be altered by natural events or by artificial means. So the resulting mutations may or may not affect an organism. So some mutations that affect individual organisms can also affect a species or even an entire ecosystem. And many of the mutations are produced by errors in genetic processes. An example, some point mutations are caused by errors during DNA replication. The cellular machinery that replicates DNA inserts an incorrect base roughly once in every 10 million bases. Small changes in genes can gradually accumulate over time. Stressful environmental causes may cause some bacteria to increase mutation rates. This can actually be helpful to the organism since mutations may sometimes give such bacteria new traits, such as the ability to consume a new food source or to resist a poison in the environment. And some mutations arise from mutagens, chemical or physical agents in the environment. And chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, and environmental pollutants. So physical mutagens include some forms of electromagnetic radiation, such as X-rays and ultraviolet rays. So if these mutagens interact with DNA, they can produce mutations at high rates. Some compounds interfere with base pairing, increasing the error rate of DNA duplication. Others weaken the DNA strand, causing breaks and inversions that produce chromosomal mutations. Cells can sometimes repair the damage, but when they cannot, the DNA base sequence changes permanently. So the effects of mutation on genes, they're going to vary widely. Some have little or no effect. Some produce beneficial variations. Some negatively disrupt gene function. And whether a mutation is negative or beneficial depends on how its DNA changes relative to the organism's situation. Mutations are often thought of as negative because they disrupt the normal function of genes. But without mutations, organisms cannot evolve. And because mutations are the source of genetic variability in a species. So some of the most harmful mutations are those that dramatically change protein structure or gene activity. And the defective proteins produced by these mutations can disrupt normal and biological activities and result in genetic disorders. Some cancers, for example, are the product of mutations that cause the uncontrolled growth of cells. Okay, sickle cell disorder, and we talked about this in class earlier in the year, is a disorder associated with changes in the shape of red blood cells. Normal red blood cells are round. Sickle cell appear long and pointed. Sickle cell disease is caused by a point mutation in one of the polypeptides found in the hemoglobin, the blood's principal oxygen-carrying protein. So among the symptoms of the disease are anemia, severe pain, frequent infections, and stunted growth. But there are some beneficial effects. Some of the variation produced by the mutagens, I just have a hard time with that word, can be highly advantageous to an organism or a species. So mutations often produce proteins with new or altered functions that can be useful to organisms in different or changing environments. So, for example, mutations have helped many insects to resist chemical pesticides. And some mutations have enabled microorganisms to accept a new chemical into the environment. Plant and animal breeders often make use of good mutations. An example would be when a complete set of chromosomes fails to separate during meiosis, the gametes that result may produce a triploid or a tetraploid organism. And the condition in which an organism has extra sets of chromosomes is called polyploidy. And polyploid plants are often larger and stronger than diploid plants. So important crop plants, including bananas and limes, have been produced this way. Polyploidy also occurs naturally in citrus plants, often through spontaneous mutations. Okay, see ya.